Oh, you finished both of them yesterday?
Because the bar was in his office, so like he was free. So.
Oh, you went to his office?
Yeah. Right.
No, I haven't. You're sitting at half microwave. We're literally opposite.
They cancel it or something? Yeah. I also have 2 more.
No, they're waiving it, so I'm.
You don't have to take it. It's actually a pretty fun class. Yeah, I actually learned that. Well, it's more like a, I feel like it's just.
That's fair. Questions?
So, what it does is it just downloads this mapping set on your phone to make your images. And then also, use like the mapping system.
Slide it under the door. I've been in it too long. That's still snow. Still snowing. I don't think I can do that. Alright, guys, I'm gonna pass around the sign-in sheet so that I can give you participation points for the worksheet. I'll start in the back, it'll make its way up to the front.
Yeah, I'm skinny like that. I'm just skinny like that.
You're not writing it with P.
Right. The other one I didn't. No, honestly.
Today's orange.
I always use orange.
I used orange on really good. It was like light gray and orange and like charcoal. And I was like, and then like purple.
Halloween.
Yeah. But like,
No. When do you do your meetings?
after my bite. Literally. I just like, don't want to get like.
I don't think you will. I've heard that. I think it's not. We're talking about Martin on the 18th. You're telling Emily and Brittany about me.
They're probably like, let me get sick of that.
Yeah, we should have been serious.
Oh. They're probably like, oh, we're all like practice. Let's take your practice. Yeah.
Just turning. We should have that too. It's just how it is.
All right, guys, while that sign sheet's going around, let's go ahead and get started. We'll go through the questions one by one like we usually do, and then I'll open the floor for you guys to ask me any questions on the lectures for the exam, which will be on Monday, April 1st. It's not an April Fool's joke.
That would be good.
I thought about that, but so unfortunately, I don't have the time to do that. But it is, less material. It's 4 lectures this time, and I think it's, a little bit easier material to digest. A lot of it, I will say, is new terminology, so, there's going to be more memorization this time and definitions and things like that on the exam, so I can promise you with confidence that this exam will be easier than the last one because of the content and just the way that I'm writing it. So this time around, there will be like true and false, there will be multiple choice, no short answer or anything like that, but like true and false is going to be added on there and like matching terms to definitions, so those are things you should focus on. So, all right, any questions about that? Okay, let's go through worksheet 6. So, these worksheets, like, I'm giving you these and they're pretty straightforward in terms of like, I'm asking you something that could be directly pulled from my slides. So I hope that you guys aren't just kind of like mindlessly filling in, these answers, and you actually are understanding the concepts behind them, because, it's kind of to reiterate these concepts, but to make sure you guys understand them and ask the right questions if you have them. So, so in the figure below, I apologize, it's not in color, but it is labeled, what is the conclusion drawn about the composition and diversity of the human microbiome? Yeah.
I said like generally more overlap, or like closer proximity is more similar, and then further apart's more different. So I think in this one, the skin and the urogenital that are most similar.
Yeah, absolutely. Exactly. And so kind of like an all-encompassing conclusion based on like all of this is that the oral microbiome is going to kind of cluster in the same place on this PCA plot, and the skin is going to cluster the same between all these individuals. So the diversity and what the microbiome is composed of is strongly determined by the habitat or the niche of that microbiome. So between individuals, everybody's skin microbiome, if I were to sample everybody's, skin, gastrointestinal tract, oral tract, and so on, you would probably bet that it would look something like this, where everybody's GI tract microbiome is going to be more similar to each other than each other's. But yeah, exactly. You can also draw conclusions like that, which are important to know. The skin in the urogenital tract here are shown to be more similar than the skin and the gastrointestinal tract, for instance. So it's important to understand that things that cluster together are more similar. All right, number 2, our microbiomes are impacted by a variety of things. List 5. There's a bunch that we listed on the board last week. So what are some things that we've talked about that can impact our microbiome? Yeah.
A few examples I put was vaginal, intestinal / fecal, breast milk, and skin microbes.
Yep. So are you talking about number 3 or 2?
Oh, sorry, 2.
Yep. You just answered 3, which is fine.
Sorry.
That also applies to 2, so it also works. Maternal microbiome definitely will contribute to impact things like that. So what else can impact our microbiome? Yeah.
Allergies, diet, hygiene, pets.
Yep. Allergies, diet, hygiene, pets, sleep. What else? Let's just kind of list them off. You can just shout it out. Thank you.
Birth method.
Birth method, birth mode, feeding mode, antibiotic use. Can't hear you.
Exercise.
Exercise, yep. Illness, sorry. Environment, sleep. Your sleep habits will definitely influence your microbiomes. Remember, your microbiomes are linked to your brain health, so if you get enough sleep, it's going to impact you really all around, and just having a good routine. Allergens, diet, all those, yeah.
Or like certain types of supplement use, maybe?
Yeah, absolutely. That would fall into diet, for sure. Yeah, because there are supplements you can take to help enhance your microbiome. That was the example I gave with the resistant starch supplementation, where the students in the class were given a resistant starch supplement. So it's resistant, which means that we can't break it down, but the microbes in our gut can, and that starch is a fiber. So it's metabolizing the fiber, and then you get short-chain fatty acid production as a result, so that can influence your microbiome. Yeah. All right. Any other questions? All right, so critical microbial exposure happens at birth. What are some examples of the maternal microbiome? So what are some aspects of the maternal microbiome that can be passed on to the baby? Either when it's in the womb, or while it's being born, or just after birth. All those things can contribute, yeah.
Microbes in the veggies now, the intestines or feces, breast milk, and just skin microbes.
Exactly, yep. All right. So I kind of just answered this, but let's go through 4. What's the difference between accessible and resistant starch?
Accessible starch is cleaved by human amylases, and resistant starch is not. And then resistant starch is a crystalline and a starch.
Yep, exactly. So accessible starch is something we have access to, our saliva. We have proteins that can degrade it and break it down, and so we are able to break it down. Resistant starch, we can still ingest it, but it's we can't break it down with our own amylases. We have certain microbes that are great for doing that, and once they do that, they metabolize it. It's food for them, and they then can produce things like byproducts and metabolites. And the example I gave was the short-chain fatty acids that are linked to promoting health. And so, kind of going off of this, during this colonic fermentation, it's important that you guys recognize that when I'm talking about resistant starch and these microbes in the gut, and fiber metabolism and colonic fermentation, those are all the same idea. They're just different ways to talk about it. So during colonic fermentation, the microbes break down the plant fibers or resistant starches. So what does this result in? It's, like I said, short-chain fatty acid production, but what are the 4 that we talked about?
Butyrate, propionate, lactase, and acetate. Acetate, sorry.
Yep. So butyrate, propionate, lactate, acetate. Those are the 4 that have been mostly studied. and have links to some health benefits as a result of anaerobic fermentation in the gut by these microbes. And so what are some health benefits of fermentation and fiber metabolism within the gut microbiome? How does it influence the epithelium health is kind of what I'm asking here. Yeah.
It increases epithelium health and function, and it decreases inflammation.
Yeah, exactly. So these are kind of, generic when we say increase epithelium health and function. It's not very specific, and that's because there's still a lot of research going on that kind of, where we're trying to conclude what these short chain fatty acids do, but there are links to, the intestinal epithelial cells in your epithelium. They need energy to be able to replenish themselves and stay healthy. And so it's been shown that these short chain fatty acids are a source for that energy that they need. So that helps with the epithelium health and function. And yes, they definitely have been shown to decrease inflammation. But butyrate or butyric acid is one of the short chain fatty acids that has been studied, I'd say a fairly amount more than the others. And so there are more specific links to health benefits. And so what are some of those health benefits of the production of butyrate? Yeah.
It decreases inflammation and likelihood of cancer, as well as the incidence and severity of GVHD.
Yeah, exactly. And butyrate in particular is the preferred energy source for the intestinal epithelial cells. So that's one of those ones that we know, it's really linked to intestinal epithelium health and function. Yeah, and then there's links to decreased inflammation, decreased likelihood of cancer, reduces incidence of autoimmune diseases like graft versus host disease, where your body will start to attack itself. There are also links to things like, I haven't mentioned this in lecture, but obesity, and things like it helps reduce obesity and things like that, because your microbiome is associated with things like obesity. All right, any questions about the colonic fermentation part?
So the four that we listed, like the butyrate, those are all short chain fatty acids?
Yes, those are all short chain fatty acids. We call them butyrate and propionate, lactate, acetate. That's the conjugate base. So technically it's not an acid. The acid is butyric acid, acetic acid, lactic acid, and propionic acid. But that's just the way that we term them. We just talk about it more by the conjugate base. But yes, those are acids.
And then those are broken down by the microbes?
Those are broken down by those microbes. Those are produced by the microbes when the microbes break down those plant fibers or the resistant starch. And I can pull that image up that kind of describes it after we go through these, and I can explain that again, just remind me. All right. So adding antibiotics to an established microbial community disrupts what mechanism? I've asked this 70 times, so you guys can just shout it out all together. Thank you. I hope you never forget that. What's another word for colonization resistance? Competitive exclusion. Good job. All right, cool. So the intestinal tract consists of both the small intestine and the large intestine, which is also called the colon. When I was an undergrad, nobody said that, so I thought the colon and the large intestine were two different things, but they are the same. So yeah, so what are some distinguishing features of both? Let's talk about the small intestine first. How is it organized? Yeah.
So it has a mucus layer, O2, antimicrobial peptides. It regulates bacterial colonization, immune epithelium. Its bacterial contact with the Peyer's patches facilitates immune sampling of microbiota.
Yeah, exactly. So in the small intestine, is the mucus layer thin? Is it thick? Is it dense? How does it look? in the picture, and it might be easier to look at the actual graphic. It was thin, yeah. So yeah, it was a really thin layer, and so that's one of those things that you should know as a difference between the large intestine and the small intestine. The small intestine is organized by this very thin mucus layer. There's oxygen and antimicrobial peptides that are there, and everything that was said already. There are these Peyer's patches that facilitate immune sampling. What about the large intestine? Yes.
It has a dense mucus layer.
Okay.
And then the outer mucus layer is colonized by many bacteria. And the oxygen is highest towards it.
Yep. Yeah. So the large intestine has two mucus layers, so that's a difference. And there's the inner mucus layer that's really dense. There is a purpose for this. It protects something. What does it protect? The epithelium. So you guys should know that the function of that dense inner mucus layer protects the epithelium directly, because it's like the inner part of the whole layer. So it has really important things or important functions in protecting the epithelium. The outer mucus layer is also protective, but in a different way, where it's colonized by bacteriophage, which are viruses that kill bacteria, and immune factors and things like that. But the inner mucus layer is right on the epithelium, so it protects the epithelium directly. And then in the lumen, there are microbes that can, that are up there protecting. They can attach to food particles, allergens, debris, and inhibit those from getting through. And yeah, like you said, oxygen is highest towards the crypt, and there are stem cells that are deep in the crypt that replenish the epithelial layer. What are the 3 microenvironments that distinguish different skin surfaces? Yeah.
You got the dry skin, the moist skin, and the sebaceous skin.
Mm-hmm.
And their environmental factors create differences.
Yep, exactly. They're environmental factors. All right. Any questions on any of that? Okay. Number 11. What are the components that make up the upper respiratory tract versus the lower respiratory tract?
Upper respiratory tract is composed of sinuses, nasopharynx, strings, oral cavity, pharynx, whereas the lower respiratory tract include trachea, bronchi, and lungs.
Yeah, exactly. Okay. So we talked about the healthy lung and how it's a low microbial biomass site. And so what that means is there's diversity, but it's low in biomass. So list 3 reasons that contribute to this. What are some things that make the lung not have a huge biomass of microbes? There are diverse microbes there, but it's at a lower numbers than the other tracts, yeah.
It's an aerobic system, there's little food, and it has a lower surface temperature.
Yeah, exactly. Those are 3 things. I'm just gonna list them all. It is aerobic, so it is exposed to oxygen. Obviously, that's how you take in oxygen into your lungs. And so it's technically an external surface. It's ciliated, so cilia kind of push things out. And there's a lower surface temperature in the trachea and bronchi. There are surfactants in the alveoli. So basically, that's just some some lipids that it's formed to kind of keep things out as well, antimicrobial peptides, very little mucus in the alveoli, and it's home to phagocytes. Phagocytes are like macrophages and immune cells that patrol the external surface and keep things out. So that's why it is a low biomass site. Any questions on that?
Where the low surface temperature was?
Yep, in the trachea and the bronchi. So more of like the lower respiratory tract. And you know, if you think about it, there's got to be little mucus in that area, because if, you know, when you get sick and you have like a lower respiratory tract or even upper respiratory tract infection, you have your sinuses building up, and then you start to build mucus and things like that. So in the lung, that's your immune system trying to build more mucus to kind of push out and try to protect itself so that, you know, more microbes can't come in and invade. So that's why normally it won't have a high mucus layer. And so when you get sick, you'll have that mucus coming out and that phlegm coming out. So that's kind of, you know, the theory behind that. And so that's different from like the gastrointestinal tract in terms of it, the large intestine having a really dense mucus layer to protect that's already there, because it's more of an internal surface. It's not so external. All right, so what are two ways of microbial immigration and microbial elimination in the lungs? So two each. Let's do microbial immigration. How do microbes immigrate to this area in the lungs?
Yes. Microaspiration and inhalation of bacteria. And that's for immigration, and for elimination, cough or innate and adaptive host.
Yep, exactly. Immigration, microaspiration, inhaling bacteria, direct mucosal dispersion is the other one. Elimination, you can cough, you have mucociliary clearance, so the cilia, you know, you're producing mucus and stuff, so they kind of try to clear things, and your immune response, your innate and adaptive host defenses. All right, what does the loss of commensal bacteria in the vaginal tract lead to?
Need to increase the microbial diversity and change the breathing and epithelial homeostasis.
Yep, so commensal bacteria exist there naturally and they don't cause harm. They're there to, you know, just exist and protect, and that's their niche. And so when you lose those commensal bacteria, that's going to kind of open up space in that niche for more microbes to come in. And in this case, in the vaginal tract, you don't want microbial diversity because potentially, you know, they're not going to be commensal organisms. They're just going to, you know, be pathogenic. And then when you have that increase in microbial diversity, then that, you know, changes the immune response, the epithelial homeostasis. It's not going to be a homeostatic environment anymore. Does that make sense? Okay. And so like we've been saying previously, having a diverse microbiota is associated with having a healthy microbiome altogether. And that's more in the context of like the intestinal microbiome. But this isn't necessarily the case for women who have which type of vaginal infection? Yes. Bacterial vaginosis, or BV. And one way to remember this, especially for women, is if you know, women are prone to UTIs or urinary tract infections, and that's basically when you have a lot of E. coli in your urine, and so you'll take an antibiotic to help get rid of it, and so it's going to kill off the E. coli, but it's also going to kill off other bacteria that are commensals, and so losing those will then allow for a diverse population of other bacteria to take over that niche, and having a lot of different microbes in that area will cause an infection essentially, and that's called bacterial vaginosis. It's just having a lot of bacteria there that wouldn't naturally be there, and they contribute to just the symptoms of what a BV infection would be. So another example that I've talked about before when taking antibiotics for UTIs is that can also then become a niche for yeast to overgrow, and so that will lead to a yeast infection. And so that's another thing of like when you're losing the commensal bacteria, you're allowing other diverse organisms to overgrow, and when they do, that's not going to be good for the host. Okay, define the gut-brain axis and the vagus nerve.
The gut-brain axis is a bidirectional link between the central nervous system and the eccentric nervous system, which is like your gut. And then it has direct and indirect pathways between cognitive and emotional centers in the brain, and then also your intestinal functions. And then the vagus nerve is an afferent, which is like going to the brain, so sensory neuron that goes from your intestinal tract up to the brain.
Yep, exactly. The gut-brain axis is this bidirectional link between the central nervous system and the enteric nervous system. And so, yeah, exactly. There's both indirect, direct pathways that, are affected. And the vagus nerve, yep, it's a, afferent spinal and vagal sensory neurons that carry feedback from the intestinal end to the brain stem. It engages the hypothalamus and the limbic system, which are responsible for regulating emotions as well. All right, what are the two routes of communication of this gut-brain axis? Yeah.
The full sensing in response to the luminal contents, and then the release of the metabolites from the GI tract to the blood.
Yep, exactly. So it can either directly sense the microbes there and locally and respond to luminal contents in the gut. And then there are also metabolites that are released by microbes, like we talked about short-chain fatty acids being one of them. But there are other metabolites as well, and that those are released from the gut bacteria. And so then they can, get into your blood. It can be systemic, get into your bloodstream and then result in subsequent effects. So it's kind of more indirect. All right. So any questions on that? All right, I'm gonna open the floor to you guys. We can go through lecture by lecture and I can answer and reiterate concepts for you. So we can start with 11, or you guys can just randomly ask me questions. Yeah.
Can you go back to the diagram for question 5 with like the butyrate and the
What lecture was that? Let's see. I think it was human microbiome. Okay, so resistant starches are basically derived from plant particles. And so that's why when I say plant particle or plant fibers, that's the resistant starch aspect. It's derived from here. And so basically this plant particle or fiber is able to be degraded. There are primary degraders, and then there are secondary degraders. You guys don't really need to know the ins and outs of these, but it's really these secondary degraders. Like primary degraders will break down the plant fibers, and then the secondary degraders can then break it down even further into short chain fatty acids. And so some of those are, these 4 here, but importantly, what we need to understand is it's these dietary fibers that come from plant particles that are food for the microbiota that can be that they can break down, but we can't because they have the specific enzymes to break them down. And then that breakdown will then result in short chain fatty acid production, which has health benefits. Short chain fatty acids have a lot of health benefits, one of them being that it can be then absorbed and then increase the tissue health through their absorption. Does that help a little? And so all of this is happening in the colon. This doesn't happen in the small intestines. It has to go all the way down to your colon. And so that's why when we can measure the levels of short chain fatty acid production, like in the study I showed you over here, the way we measure levels of butyrate in each of these individuals is by, sampling your feces, and that's coming from your colon, because that's where things are being broken down. So it's also important to know where it's happening. What else?
For the bacterial classification in lecture 11,
Yeah.
you have to know like each domain by what class, or like a certain for that bacteria, or
No, generally, but what you should know is, like, if I were to say, if we're talking about E. coli, and I tell you we're talking about Enterobacteriaceae, which classification would that fall under? You should know that it's the family. And so, yeah, and then the genus would be Escherichia, species would be coli. And what is the domain, the bacteria? So it would be important to know the ones that are listed here, the Lactobacillus and the Escherichia ones.
So with the intestinal tract, the small intestine, large intestine, it asks like what are some distinguishing features.
Yep.
I was kind of looking through the pictures and I was naming like the types of cells and stuff.
Mm-hmm.
Why don't you kind of go over some of them?
Yeah, absolutely. All right, so let's go through here. Also, this one would be an important one to look at here. So, so this is the stomach. It's attached to the small intestine, which is composed of the duodenum, jejunum, and ileum. So the small intestine is all like here in the purple. And then the colon is the large intestine, and so it's over here. And it's really split into the proximal and the distal colon, which is just the top part and the bottom part. It's attached to the rectum, and so the feces can then, go through this way. And so if we go over here, it's important to also know, like when we're distinguishing the small intestine and the large intestine, you don't necessarily have to know like the species that are mostly predominant in those areas, but one thing that you should know is that as you go down this whole system, you have more bacteria. There are more microbes in the large intestine than there are in the small intestine than there are in the stomach. And so this is kind of how it's split. And so as you go down, there's more bacteria, but the oxygen is going to be decreasing as you go down, because remember that fermentation, that colonic fermentation that's happening in the large intestine, those are anaerobes. It's strict anaerobes that are producing short chain fatty acids. So they don't like oxygen. They don't survive with oxygen. We've evolved to keep oxygen away through, facultative anaerobes using up the oxygen so they can survive. So as bacterial load goes up, oxygen levels go down. And then pH, you don't have to know much about, but you should understand that the mucus type and thickness is also going to increase in the large intestine. And so it's got to be really protected because there's a lot going on there. So yeah, you should know the bacterial load, the oxygen, like where it's highest and lowest, and then the mucus type and thickness. And so if we're looking at just the small intestine, so here we have our, can you see this? Oh yeah, you can, but I think it might be easier for me to use my pointer. Okay. So in the small intestine, you have these are your intestinal epithelial cells. They consist of just things called Paneth cells, stem cells, goblet cells that produce mucus. You don't have to know the different cell types that make up the intestinal epithelial cells, but these are just names for them. And so it's protected by a very thin mucus layer. You can see how, like, if you go down, it's going to be a little bit more mucus here, but in general, the things that are protecting it from being invaded and getting a small intestine infection is this mucus layer, oxygen, and just antimicrobial peptides. So microbes that are helpful and are commensal will produce peptides to kind of defend and keep things off from invading. So all these are at work together to kind of regulate the bacteria that's colonizing and organize what's going on near the epithelium. And so we've seen that there are these Peyer's patches. They're just these little patches of cells that are where there are basically immune cells below over here. And so if there is something going on up here on top of the intestinal epithelium, this is a bacteria here, this cocci that's in a chain, is trying to get in and trying to invade the epithelial layer here. And so it's this contact over here at this area that we're calling a Peyer's patch that then facilitates this immune cell, this dendritic cell, to recognize, there's something trying to come in, and so this will then come over here and try to sample, what's going on. So that's what that Peyer's patch is. There's also a little area right here where you see it also trying to do the same thing. Alpha defensins are an example of an antimicrobial peptide. Does that help a little? Yep, okay. And then, and then just to note here, the oxygen level is going to be highest up here in this area, and then it's going to be a lot less over here. But that's not going to matter too much. What you should know is how it differentiates between the large intestine. And so for the large intestine, it's also regulated, what's going on over here, the microbes that are going on over here is still, regulated, but it's regulated differently. It has this dense inner mucus layer, and so because it's directly contacting these epithelial cells, you'll see that it just directly is trying to protect this epithelium. And then there's another mucus layer that's there too, and this mucus layer is protecting by also containing and being colonized by just a lot of different bacteria that are commensals, bacteriophage, so viruses that kill bacteria, and just immune cells and immune factors. So also some antimicrobial peptides that are being secreted, but that's all in the outer layer mostly. And so those are all just there and trying to keep things, you can see here a phage is trying to attack a bacteria that's trying to come in. If there's a food particle, so this part is the lumen, so if there's a food particle that's trying to come in and say it's something that the person is allergic to, there are bacteria that are going to come and try to attack it, and so in the lumen, microbes can attach to food particles and debris, so they're also protecting. So there's a lot of added layers of protection in the large intestine compared to the small intestine, and that has to do with oxygen being lower, having a lot more microbes in that area, so there's a lot more going on, a lot more mucus being produced. Oxygen is normally highest towards the crypt. This is the same. This is also similar for the small intestine. And then deep in the crypt, there's stem cells as well that help replenish it. But you should understand the concepts of what the inner and outer mucus layer are doing, and why there's so much more bacteria in this area compared to the small intestine. Higher biomass. Does that help a little? What else?
Was the higher biomass due to like partially the oxygen too?
Yeah, because there's lower oxygen, there's a lot more, and like pH and all of these things kind of all together are going to contribute to why there's a higher biomass. But yeah, we've evolved to have a lot more microbes in the large intestine that are commensal, and that's because the bacterial load is higher because there's a lot less oxygen, higher pH. So higher pH means it's more basic. And so one thing you can think about is the stomach has the lowest pH. And so the stomach is very acidic. And so a lot of things that you eat that go through to the stomach kind of like get destroyed because it's such an acidic environment. So there's this whole thought of like, for instance, this is me going off on a tangent, but probiotics, you take a probiotic and you're hoping that what you're hoping when you take a probiotic is that it gets through the stomach, through the small intestine, through the large intestine, and it's feeding those microbes that are supposed to be good for you, and you're supposed to feel better and have better health effects. But one issue with some probiotics is you eat them, they have to go through the stomach first, but then they get destroyed by the acids in the stomach before they even reach the small and the large intestine. So, that's why the pH is a lot lower here. And that is also going to contribute to the mucus thickness and things like that. So less bacteria can survive the acidity of the stomach. So once you have more basic pH, the bacteria are able to survive that. And so that's why there's higher bacterial load. So these are all really connected. Did everybody sign in, by the way? What else?
For that image, like I get like the mucus type of thickness, like it's getting thicker as you move further down. But like, what does it mean by like the type? Just like, is that what it means? Just generally the
Type and thickness. I don't worry too much about that. I think what it's talking about there, they're just being specific because in the colon there's two types, the inner and the outer, and then what's contained in them, like the outer one is less dense, but there's a lot more microbes and immune cells and stuff in that mucus layer. That's what they mean by type, but it's nothing you guys need to be worried about. Yeah, good question. I didn't even notice that at all. What else?
What are some of like the higher yield stuff like true and false? Because I know like definition wise.
Sorry, can you say that again?
The true and false questions you said that were gonna be on there, like, Are there like more summer topics that we should focus on more for those kind of things?
I guess, you know, just kind of going through and knowing like the terms like, I don't want, a lot of it is like you have to understand the concepts, but memorization is a part of it too, you know, like understanding it, like true and false are gonna just kind of be parsed out. Like, let me think of something. Like I could ask you, you know, the inner mucus layer is colonized by many bacteria, macrophages, or bacteriophage and immune factors in the large intestine, true or false? It's false, because it's the outer layer. So things like that. Like direct, you know, statements that I've made, and it would be, you have to really just like know the little differences there. Are you guys good on the richness and evenness? The microbial ecology, the symbiosis, you know, understanding the differences between them. What else? So when it comes to the three microenvironments of the skin, you know, we said the composition of them is influenced by environmental factors and host factors. But one of the ones that I wanted you guys to remember is that in sebaceous skin, you can see here that it's about 50% of the skin is colonized by Propionic bacteria, because this is the contributor to like acne, same as corny bacteria, but Propionic bacteria is one of those that really contributes to acne. The bacteria that causes acne is called Propionus acnes, so that's one that you should know as a difference. I don't think I was specific about that in the worksheet. And then, yeah, just understanding that context matters in these relationships between the microbe and the host. Different microbes colonize different different habitats in the oral cavity. The oral microbiome can impact the rest of the body, remember, and we also talked about obviously the gut-brain axis and how the gut can influence the rest of the body, but I know I also, you know, briefly talked about the oral microbiome impacting other areas of the body like cardiovascular disease, Alzheimer's, inflammatory bowel disease, rheumatoid arthritis, so a lot of different things. You guys understand this concept of ecological succession? Yesterday we did this experiment where we're using coliform bacteria, which are bacteria that are coming from the colon, as an indicator or organism for water quality. And so that would be looking for those gram-negative lactose fermenters, E. coli being one of them. So here we talked about ecosystems with higher species diversity tend to be more resilient. You know, this is definitely true, but I do want to reiterate there are exceptions to every rule, like I've said before, and this is not the case, for instance, in like the vaginal tract, where if you have diversity, it's not going to be good for the host. You're losing those commensals, and so that's going to elicit an imbalance and loss of homeostasis. So, but this does definitely apply to other ecosystems, including environmental ecosystems, your gut ecosystem, and things like that. Knowing the difference between alpha and beta diversity. In this case, in the skin microbiome, diversity is also really important for human health. Decline in biodiversity results in not having a lot of microbes. They're being deprived, there's an imbalance, there's a disturbed immune response or dysbiosis. You have a higher risk of inflammatory diseases. Can we know everything about an organism just based on culturing it in a lab? No, exactly. That could be like a true and false question.
Isn't it like you can only like say something's not there by culturing it in the lab? Like you can determine that.
Yeah, so where did we talk about that? Lecture 12. Okay. Oh, there it is. That's for enrichment cultures. So if you're trying to enrich for an organism alone, so if I give you a sample with a mixed culture and I want to try to just isolate out like a certain bacteria, I try to enrich for just that. Like I put in this media the specific things that that organism would need that would allow it to grow over the others in that mixed culture. And so if it does grow, you can prove that it's there in that habitat. If it doesn't grow, you can't say that for sure it's not in that habitat. Maybe you just didn't have the right, stuff in there. And so the ability to be able to isolate it from an environment says nothing about its significance in that environment ecologically. So yeah, so basically you might not know the exact requirements in the proper form and culture. And some things that might lead to that is like, either you waited too little, like that you needed more time to let it grow, or you have the wrong pH. Or a really important thing is in the environment, it's growing with these syntrophic partners. So maybe you're missing those partners in the lab setting, but it needs to be there. Maybe we just don't know that yet. So there's so many things that can lead to it. So even if it doesn't grow in an enrichment culture, you can't prove that it's not there in the environment that it's supposed to be in. And if it does grow, then yeah, you can say, okay, yeah, it is there. Does that make sense? Mm-hmm. Good question. I'm not gonna like hit you hard with questions about these. Just know like the key things, like for PCR, for instance, you need these things for PCR, and then the one that isn't in here, remember I said, is the DMTPs, so the A's, C's, T's, and G's that you'll need to put in there as well in the mix to be able to add those, when the primer is elongating the strands. Heating and cooling, like I've said before, is not an ingredient. The bullet point should have been indented out. What else? You guys understand the difference between a population and a community? You should understand the two-way relationships, one-way relationships, negative interactions, positive interactions, examples of them, things like that. Those are mostly memorization, but you should be able to understand them conceptually, ones that can live together, that can't live independently, and ones that can live independently. I've shown you these two slides before in a previous lecture. Examples of mutualism, you should understand these examples and what's happening. That's why yesterday I kept saying paramuseum when I was trying to say parathecium. I was like looking up the slide. Competition usually leads to excluding a less successful population. Parasitism versus predation. Keystones. What else? Say that again?
Do you want us to know like this whole summary of the sequencing? Or?
The sequencing? No, you don't have to know the whole thing, no. No. what you should know is it a culture-based method, is it culture independent, and that kind of thing, like the very overall concept. You don't have to know the details of it. And like, what are they using? DNA, that kind of thing. Anything else? We have 15 more minutes, so I can stay here and you guys can ask me questions. Thank you. Yep.
Can you explain how a strain can be like the same or different?
Yeah, so, let me go back to right here. Okay. So an isolate is essentially like you just having a single colony. And so each one of these is an isolate. So they can all be the same bacterial strain. It can be like the same thing that you've isolated essentially. But having two different strains of something, then they're gonna be two different isolates. Yeah. So two isolates can be the same species or they can be different. Yeah, or they can be the same, but they, two isolates of the same species can be two different strains, or they can be the same strain. The two different strains of the same species are gonna be two different strains, so they're automatically two different isolates.
Okay.
Yeah. That part can get confusing.
Have a good day. You too, thanks.